I want you to imagine, just for a second, that you are trying to study the engine of a car.
Okay, a car engine.
Right.
Yeah.
And you need to know exactly how the pistons fire, how the spark plugs connect, like every single microscopic mechanical detail. But there is this massive catch.
There's always a catch.
Exactly. You can only look at this engine while it is entirely submerged in a vat of thick black motor oil.
Oh, wow. Yeah, that would obscure pretty much everything.
Right. And it gets worse. If you try to pull the engine out of the oil to get a clear look at it, it instantly shatters into a million useless pieces.
Well, I mean, that's an impossible engineering puzzle, right? Because the very environment that keeps the machine functioning is exactly what obscures your view.
And for you listening right now, until literally last week, this is an impossible problem in biology, too. Scientists face this exact dilemma with something called membrane proteins.
Yeah, and we should clarify, these aren't just like some niche scientific curiosity.
No, not at all. They are the target of more than half of all clinically approved drugs. Like, if you're taking a medication right now, there is a coin flip chance that it interacts with one of these impossible-to-study biological engines.
Which makes this topic so incredibly vital.
It really does. So our mission for this deep dive today is to unpack a brand new paper that just dropped in the journal Science. It's titled Membrane Protein Solubilization and Structure Determination Using De Novo Designed Proteins.
It's quite a mouthful, but the implications are, well, they're staggering.
Yeah, basically they used AI to invent a microscopic spacesuit to pull these biological engines out of the oil completely intact.
Right, which fundamentally changes how we approach drug and vaccine discovery. I mean, we are moving from a paradigm of basically destroying these proteins to perfectly preserving them in plain water.
It's wild. But before we get to the whole AI spacesuit thing, let's establish why these proteins shatter in the first place. So I know they sit inside the cell membrane, which is basically a wall of fat that separates the inside of the cell from the outside world.
Exactly, a lipid bilayer.
Right, but why can't we just, pluck them out of that wall to study them?
Well, it really comes down to amino acid chemistry. So proteins are these long chains of amino acids that fold into really complex three-dimensional shapes.
Like tangled up yarn.
Yeah, like yarn that has to be in one very specific knot to work. And for a protein embedded in a lipid bilayer, that wall of fat, the amino acids on its outer surface have evolved to be highly hydrophobic.
Meaning they repel water.
Right. They chemically repel water. They love the fat, but they have a phobia of water.
So if you drop them into a standard water-based lab solution to study them, what actually happens? Do they dissolve?
Actually, they do the exact opposite. They essentially panic.
They panic.
Yeah, on a chemical level. To minimize their exposure to the water, the hydrophobic surfaces of multiple proteins will violently stick to each other.
Because they're trying to hide from the water.
Exactly. The protein misfolds, it aggregates into this useless, clumpy mass and completely loses its structural integrity.
Okay, that makes sense. It's aggressively trying to hide its water-hating parts. Now, I know the traditional workaround for this in the lab was using detergents, which is essentially soap, right?
Right, yeah. Detergents.
But that always sounded so counterintuitive to me. I mean, it's like washing your greasy hands with harsh dish soap, but the soap actually dissolves your fingers in the process. If I'm trying to preserve a fragile biological structure, blasting it with industrial soap just seems like a terrible idea.
Well, that's a really common misconception, actually. In biochemistry, detergents don't really blast the protein.
Oh, they don't?
No, the detergent molecules have this hydrophobic tail and a hydrophilic or water-loving head. So they surround the protein, hiding its hydrophobic parts inside this little protective bubble.
Okay, wait, I think I've heard of this. Is that called a micelle?
Yes, a micelle.
Okay, so if it forms a protective bubble, then why is this new science paper treating detergents like they're the ultimate enemy? That sounds like a pretty elegant solution to me.
Right. Well, what's fascinating here is that it's like trying to remove a stain from a really delicate silk shirt using a high-pressure power washer.
Right.
You might get the stain out, sure, but you shred the fabric in the process. Those detergent micelles are highly dynamic. They're squishy.
Squishy isn't great for a structural shield.
Exactly. They constantly slosh around, which actually strips away the vital natural lipids that hold the protein together. So over time, the protein just loses its stability and falls apart anyway.
So you manage to get it out of the membrane, but you slowly destroy the exact machine you're trying to study.
Precisely. And plus, if you're trying to take a high-resolution photograph of this protein to, say, design a drug to fit perfectly into it.
Right. Having a squishy soap bubble constantly moving around the target seems like it would just blur the image.
It absolutely does. The micelle masks the true structure. It adds this thick layer of visual static to the image.
And I read that previous workarounds involved genetically mutating the protein to force it to like water, but that literally changes its true nature.
Yeah, or fusing it to generic floppy proteins like EpoAI, which just makes them too flexible to get a clear structural picture.
Okay, so let's do a quick recap for you listening. The problem is that membrane proteins hate water. They clump up and die. The old solution was soap bubbles. But soap bubbles strip away the structural integrity and blur our imaging.
Which means we desperately needed a new approach.
Right. We need a way to protect the protein without chemically assaulting it, like a bespoke spacesuit tailored for every individual protein. And that brings us to the star of this paper, the WRAP.
Yes, WRAP.
Right, which stands for water-soluble, RF-diffused amphipathic proteins. Let's give our listeners a quick mnemonic for that. Amphipathic essentially means having a split personality. For like amphibian, right? Living in two environments.
That's a great way to remember it. One side loves water, the other loves fat.
Exactly. So what physically is a wrap?
The wrap is a genetically encoded, custom-designed protein shell. So the inner surface perfectly hugs the greasy, hydrophobic parts of the target protein,
Making it feel like it's still in the fat wall.
right. While the outer surface presents this very friendly, hydrophilic face to the water around it. And the best part, absolutely zero detergents are required.
Okay, designing a custom microscopic shell sounds great in theory, but, proteins are incredibly complex, highly specific folded structures. They aren't just smooth cylinders. How do you design a suit for something so intricate? Do they just, guess the dimensions until a tube fits over it?
Well, historically, maybe, but not anymore. They actually use a deep learning pipeline starting with an AI tool called RFdiffusion.
Oh, this is from the Institute for Protein Design.
Yes, exactly. It's a generative AI model for protein backbones. So you basically ask the AI to generate an idealized hollow cylinder.
What's a cylinder made of?
It could be a bundle of coiled springs, which we call alpha helices, or it could be a woven basket structure called a beta barrel. The AI creates a basic tube that roughly matches the height and width of your target membrane protein.
But a generic tube isn't going to fit perfectly. It's like buying an off-the-rack suit. It might cover you, but it's going to be really baggy in all the wrong places.
Which brings us to the secret sauce of the whole paper, partial diffusion.
Okay, partial diffusion. Let's unpack this because this was really cool in the source material.
Yeah.
So they digitally place the target protein inside this generic AI tube and then use the AI to mold the tube to fit.
Correct.
I was trying to visualize the mechanism for this, and it made me think of key cutting.
Oh, okay. How so?
Like if you have a blank key and you cover it in warm, moldable clay to get an impression, you don't want to completely randomize the clay until it's just mush, right?
Yeah.
You only want to soften it just enough so it sinks into the unique microscopic bumps and grooves of the original key.
That is an excellent way to conceptualize the denoising process in machine learning. Seriously. Yeah. Because in this AI model, they typically use a denoising trajectory of, say, 50 steps to generate a completely new protein from pure noise.
Right. Full randomization.
Yeah. But for partial diffusion, they don't run the full process. They actually skip the first 30 steps and only run the final 20.
Oh, so they just soften the digital clay.
Yes. The AI slightly alters the backbone of the cylinder so it achieves perfect shape complementarity with the target protein.
Wow.
It learns the exact topography of the membrane protein's exterior without losing that overall cylindrical spacesuit shape.
Okay, so now we have a 3D blueprint of a suit that perfectly hugs our target down to the atomic level. But a digital blueprint doesn't tell a biological cell how to actually build it.
Right. Shape is just the beginning.
Yeah, how do they translate a shape into a sequence of amino acids?
That requires another tool, a sequence design algorithm called soluble MPNN.
Soluble MPNN. Okay, what does that do?
Its job is to look at that molded 3D backbone and assign specific amino acids to every single position. And it operates under really strict chemical rules.
Because it has to be amphibian-like, right?
Yeah.
Amphipathic.
Exactly. It assigns amino acids with surgical precision, ensuring the interior sequence chemically bonds with the target's hydrophobic exterior, while the exterior sequence is perfectly hydrophilic.
It's literally painting the inside with oil-loving chemistry and the outside with water-loving chemistry. That is brilliant.
It is, but before they actually spend the time and money to synthesize this in a real lab, they have to verify it.
Right, and I saw they used AlphaFold 2 here. Now, are they just using it to double-check their geometry?
Oh, AlphaFold 2 is far more than a simple geometry check. It's a rigorous physical simulation.
What do you mean?
It predicts how a linear sequence of amino acids will naturally fold into a 3D structure based on all the complex physical and chemical interactions between those molecules.
So it's basically asking, will physics actually allow this to exist?
Yes. If AlphaFold 2 predicts that the sequence from soluble MPNN will indeed naturally fold into that exact spacesuit shape hugging the target protein, then they know they have a viable design.
Okay, let's do another quick mental recap for you listening, because this pipeline is dense. We've gone from a fragile, water-hating protein stuck in a wall of fat to a purely computational AI pipeline. RFdiffusion makes the initial shape, partial diffusion molds it like warm clay, soluble MPNN assigns the oil and water chemistry, and AlphaFold 2 verifies the physics.
That is the perfect summary.
Awesome. So having established this incredible theoretical pipeline, the natural next question is, does it actually work in a living cell?
Right, time for the physical biology. To manufacture these suits, they turn to a classic workhorse, E. coli bacteria.
Good old E. coli.
They genetically programmed the bacteria to produce both the target protein and the W-wrap suit as a single connected chain.
And the first major test they ran was on an E. coli outer membrane protein called UMPA.
Right, UMPA. And the results were, frankly, striking. The bacteria produced the WR-apt UMPA directly into their watery cytoplasm, totally bypassing the fat membrane.
Because it's already in its water-soluble suit.
Exactly. When researchers extracted it, it was completely soluble in plain water, no soap required.
But what really caught my eye in the paper was the thermostability. They heated this W-apt protein to 95 degrees Celsius.
Which is nearly boiling.
Right. Most proteins unfold and cook at those temperatures, like a frying egg in a pan. But this didn't. The W-wrap acted like an armored thermal jacket.
It survived completely intact, which, gave him the confidence to tackle a much harder, infinitely more important target.
And this is where the real-world medical application comes in. We're talking about Treponema pallidum, or TP, which is the bacterium that causes syphilis.
If we connect this to the bigger picture, designing a vaccine for syphilis has been historically, it's been a nightmare.
Why is that? Why is syphilis so hard to crack?
First, TP is incredibly difficult to cultivate in a lab. But more importantly, its outer membrane proteins, which are the prime targets you would use for a vaccine, are extremely fragile.
Oh, because they're stuck in that lipid bilayer.
Yes, and they're notoriously difficult to extract intact. Before this paper, there were 0 experimentally determined structures for TP integral outer membrane proteins.
Wait, 0?
0. The scientific community was essentially flying blind.
Okay, but designing a custom shield for a protein you've literally never seen in real life feels like an incredible leap of faith. How did they build a wrap for an invisible target?
Well, they had to rely entirely on AlphaFold 2's predictions of what the TP protein should look like based purely on their genetic sequence.
Wow.
Yeah, they designed wraps for 4 different TP proteins using only those computer-generated models. And when they expressed them in E. coli, they were completely water-soluble.
That is amazing. But hang on, I need to push back on the vaccine application here.
Okay, push away.
So if the whole point of a vaccine is to train the human immune system to recognize a protein, but you've just wrapped that target protein up in a synthetic AI-designed spacesuit, doesn't the suit hide the target? Like, how can an antibody see what's covered up?
That is a very critical detail. If the W-wrap occludes the antigenic sites, it's completely useless for vaccine development.
Right. So how did they prove the targets were still visible?
To prove it, they used an ELISA test. ELISA.
Okay, let's walk through the mechanism of an ELISA test for the listener. It's essentially a biological trap, right?
This is a really good way to look at it. So you coat a plate with your synthetic W-apt TP proteins.
Okay, plate is coated.
Then you introduce blood serum from rabbits that have been infected with real, live syphilis.
So that serum is packed with natural antibodies hunting for the type of bacteria.
Exactly. If the antibodies physically grab onto the proteins on the plate, a secondary chemical reaction produces a color change, proving recognition.
It lights up, basically. And did the rabbit antibodies grab onto the W-wrap proteins?
They reacted very strongly.
Wow.
This proved that the W-wrap was only hugging the fatty middle of the protein. The critical immune targets the extracellular loops that stick out of the top and bottom of the cell membrane. They were protruding from the graft perfectly.
So they maintain their natural, recognizable shape.
Exactly. The immune system could see them clear as day.
Okay, so that solves the structural visibility problem. We have a stable, soluble protein that the immune system can actually see. But proteins aren't just static sculptures, right? They are dynamic machines.
Very dynamic, yes.
If you force a microscopic machine into a tight, custom-fit spacesuit, my intuition tells me it wouldn't be able to move. Like, can it still actually do its job?
Function over form. It's the ultimate test. And to answer that, they looked at an enzyme called GLPG.
GLPG. What does that do?
It's a protease, which means it acts like molecular scissors to cut other proteins inside the inner membrane.
Okay, but how do you measure if invisible molecular scissors are actually cutting something inside a test tube?
You use the fluorescent probe, specifically one called FP-TAMRA.
FP-TAMRA. Okay, how does that work?
This probe is designed with a very specific mechanism. It only lights up and covalently bonds to the enzyme if the enzyme's active site is currently engaging and cutting a target.
Oh, so it's like a definitive chemical flag for activity. If it's cutting, it glows.
Right. And the WRF-GLPG...
Did it light up?
It was brightly fluorescent.
No way.
Yes. The enzyme was actively cutting targets exactly as it would in nature. The suit didn't restrict its necessary conformational changes whatsoever.
That is incredible. Okay, so we've covered bacterial enzymes and syphilis vaccines. Let's bring this to human drugs. Let's talk about GPCR, G-protein-coupled receptors. The source paper mentions they WR-aped a human receptor called mini-CXCR4.
Yes, mini-CXCR4.
Now, for the listener who might not know, why are GPCRs considered the holy grail of drug discovery?
Well, GPCRs sit on the outside of almost every single human cell. They read chemical signals from the outside and transmit instructions to the inside.
They're the communicators.
Exactly. Because they are the ultimate communicators, they are implicated in everything from heart disease to cancer metastasis. In fact, they are the target for over 30% of all FDA-approved drugs.
30%. That's massive.
It is. But because they are membrane proteins, screening new drugs against them traditionally requires those destructive detergent micelles we talked about earlier.
Those squishy soap bubbles that ruin everything.
Right. But the WRAP eliminates the detergent entirely.
So did the WRAP human receptor still communicate? Did it still bind to things?
It did. It successfully bound to its natural signaling molecule, which is called CXCL12.
Okay.
And crucially, it's strongly bound to a small molecule drug called plerixafor, which is currently used to mobilize stem cells. The binding affinity was, it was around 390 nanomolar, which is excellent.
You getting it grabbed on tight?
Yes. This proves pharmaceutical companies could theoretically mass-produce stable human receptors in plain water and just screen millions of drug compounds against them flawlessly.
That would speed up drug discovery so much. Okay, here's where it gets really interesting. Let's look at the ultimate proof of flawless preservation. The final boss of this paper.
The MSPA nanopore?
Yes. This thing is a behemoth.
It really is. MSPA is a massive protein complex formed by 8 identical subunits coming together to create a central pore or a hole.
And it's widely used in biotechnology for rapid DNA sequencing, right?
Exactly.
The mechanism there is fascinating. You listening, imagine this. You literally pull a physical strand of DNA through the microscopic hole in the protein. And as the DNA passes through, it disrupts an electrical current in a very specific way that lets you read the genetic code, literally letter by letter.
It's brilliant, but it's an incredibly delicate machine. And because of its massive size and perfect symmetry, it was the ideal candidate for high-resolution imaging.
Using cryo-electron microscopy or cryo-EM.
Right.
So cryo-EM is where you flash freeze the proteins in a microscopically thin layer of ice, and then you shoot a beam of electrons at them to build a highly detailed 3D map.
And the map they generated for the WR-aped MSPA was at a staggering 2.95 angstrom resolution.
Okay, let's contextualize that. An angstrom is roughly the diameter of a single hydrogen atom.
It is unfathomably small. So at 2.95 angstroms, you aren't just looking at the general blob of the protein. You can literally map the individual twists, turns, and side chains of the constituent amino acids.
And here's the true test. They took this atomic-level photograph of the WR-aped MSPA, and they superimposed it over the known structure of the native protein in its natural fat environment.
The moment of truth.
Were they even close?
The structural difference was 0.4 angstroms.
Wait, 0.4, that's less than half an atom of difference?
Less than half an atom.
That is mind-blowing. It proves that the WRF does not crush, warp, or distort the native protein at all. It perfectly preserves the exact atomic-level architecture in plain water without a single drop of messy detergent.
It fundamentally proves the entire premise of the AI pipeline.
So what does this all mean? Let's bring this all together for you listening. For decades, roughly a third of all human biology, the gatekeepers, the infection pathways, the most important drug targets, has been locked behind this greasy hydrophobic wall.
And studying them was literally like trying to analyze that delicate car engine while it drowns in oil.
But with JobRiffs, the scientific community now possesses a plug-and-play, custom-tailored, AI-driven solution.
Right, we can pull these vital proteins into the light completely intact. It clears the path to map the outer armor of syphilis for new vaccines and to screen novel drugs against human receptors cheaper, faster, and more accurately than ever before.
And honestly, it forces us to rethink what AI is truly capable of. The next time you hear a headline about an AI breakthrough, remember, it's not just about language models generating emails or chatbots writing code.
No, this is deep learning, literally designing physical matter to solve fundamental biological bottlenecks.
AI is reshaping the physical tools we use to understand the microscopic machinery that keeps us alive.
It's a whole new frontier.
Which leaves me with a final provocative thought for you to chew on today. Have you ever wondered what the limit of this tech is? If we can now use AI to design custom, water-soluble spacesuits that perfectly hug and protect fragile membrane proteins outside their natural environment, could we eventually design therapeutic wrappers that you inject directly into a patient?
Oh, that's a wild thought.
Right, like wrappers that perfectly surround and neutralize a specific toxic molecule, or even trap whole live viruses inside a custom-fit AI cage right there in the human bloodstream.
I mean, once you can design a protein to perfectly mold to any target shape you want, the possibilities for therapeutics are virtually endless.
From an engine drowning in oil to a custom-tailored cage that could trap a virus. Thanks for taking the deep dive with us today.